Hello students, how are you all? Welcome or welcome back to my EduSpace. Hope you all are doing and learning well. If you like my videos, then don't forget to subscribe my channel and you can share it to your friends as well. So let's talk about today's video. It is in the continuation of previous lysosome video. This is gonna be the second part of lysosomes in which I'm going to tell you about the various functions of the cell survivor, that is lysosomes. So without further ado, let's get started. So before starting the video, let's take a quick review of our previous video, in which we have discussed about the etiology of lysosomes. Lysosomes means the body that lyses, or the body or the structure that are used for lysing or breaking down the larger molecules into smaller ones. Little bit about the history, that is Christian D. Dewey coined the term lysosome. How we can say that lysosomes are trash can of a cell or the waste disposal or cleaners of a cell. We also discussed about the structure of lysosomes. It is spherical in shape, okay, that contains so many hydrolytic enzymes in its lumen. So this is the structure of a lysosomes, which is spherical sac-like structure having an outer membrane which is quite equivalent to the plasma membrane, and inside which in the lumen there is a lots of hydrolytic enzymes are present. There is also one more thing, that is transport proteins. So many transport proteins are present on the membrane of the lysosomal sac, which are useful for transporting protons, that makes this lumen very acidic. We also discussed about the why lysosomes are known as suicidal bags. We discussed about the formation of lysosomes and also how lysosome works. What are the functions of lysosomes? Lysosomes are involved in so many cell processes. They not even destroy the foreign molecules that comes from the external environment, but also help in destroying the non-functional organelle, that is undergo apoptosis, which means programmed cell death, if the cell is damaged or if the cell is beyond repair. Okay, so the first function is intracellular digestion. I just mentioned that lysosomes helps in two ways. First, it destroys the foreign particle. Suppose this is a foreign particle or foreign stuff, foreign material, which may be bacteria, viruses. Okay, through phagocytosis, it is engulfed by the cell and it comes inside the cell, which is now called phagosome. Okay, this is now phagosome. This particular phagosome is got fused with our lysosome containing hydrolytic enzymes. So it gets fused with the lysosome and all the foreign material got lysed. So we can make this like the fusion of phagosome with lysosome, which causes the pathogen lysis. which is used by the cell, because now it is in its simplest form. And the other one is it also helps in destroying or lysing the intracellular components that are non-functional or beyond the repair. So suppose again this is our cell having nucleus, and suppose this is mitochondria, which is now non-functional. So this mitochondria is enveloped by the membrane of smooth endoplasmic reticulum, and then it got fused with lysosome. It becomes autolysosome, and after that this mitochondria got disintegrated with the help of hydrolytic enzymes that are present in the lysosomes, okay, which is used by this particular cell as a nutrient. So let's talk about the intracellular digestion. We know that every organism needs energy to live, okay. Cell does not only retrieve molecule but is also break it down. This is intracellular digestion. So what is intracellular digestion or process? That there is a retrieving of molecules, and then after retrieving the molecules, with the help of lysosomes, this particular molecular material get digested or break down. This is known as intracellular digestion with the help of lysosomes. It is quite analogous to our stomach, where digestion of materials or digestion of food takes place. It may be a heterophagus or autophagus. And phagus and phagine means to eat. Okay. So this is an endocytosis process where foreign stuff undergo digestion within cell. The endocytosis includes the process of phagocytosis. Phago means food, to eat, okay. It may be phagocytosis or pinocytosis. Pinion means to drink. If the foreign particle is solid, then it is phagocytosis process, and if the foreign stuff is in liquid form, then the cell used to drink it. So it is known as pinocytosis. So we can say endocytosis is such process through which foreign stuff undergo digestion. It is two types. The first one is phagocytosis, where solid material get digested, and pinocytosis, where the liquid form of stuff is digested. In phagocytosis, the phagosome containing ingested stuff fuses with a lysosome to form a phagolysosome, inside which ingested stuff is digested by hydrolytic enzymes of lysosome. As I just told you that in phagocytosis, suppose this is our solid stuff that is going to be engulfed by this cell, okay. It makes phagosome. This is our phagosome. Then the lysosome that is present inside the cell is get fused with this phagosome, and they become phagolysosome. Inside the phagolysosome, this solid stuff is digested or break down into simplest form, which ultimately get used by this particular cell. Such kind of digestion are present in those animals which lack digestive tract. Okay. Like for example, the protozoans in amoeba, paramecium, poriferans or sponges, mollusks, etc. They all used to perform such type of digestion. Intracellular digestion. It is divided into two types. The first one is heterophagic digestion and the other one is autophagic digestion. The hetero means other. Okay. Here the hetero means other, which is not the part of a cell. The other or the foreign, that is not the part of the cell. Whereas auto means self, or we can say these are the components that are the part of the cell. So in heterophagic digestion, the lysosomes break down the molecules that are brought into a cell from the surrounding via process endocytosis. So in this is a cell and this is the foreign stuff. This is get endocytized from the surrounding, and then the digestion with the help of lysosome takes place. Okay, so this is a foreign particle. Sorry, this is a foreign stuff that is going to be digested with the help of hydrolytic enzymes of lysosome. Okay. And the rest one, which is not used by a cell, is spit out with the process known as exocytosis. So this is the process exocytosis where the unwanted or waste material is spit out by the cell. So it belongs to process. The first one is endocytosis. This is endocytosis, where the foreign particle is engulfed by the cell. Endocytosis. Here is a fusion of phagosome and lysosome. Fusion. Okay. Now the next is in autophagic digestion. The lysosome digests its own internal molecules that are non-functional or infected. So in this, we already discussed this, that inside there are so many cytoplasmic organelles that may be infected or non-functional are digested with the help of lysosomes. Okay. So this type of digestion is known as autophagic digestion. This is our lysosome. And these are cellular components that are going to be digested because they are no more in functional process. Cellular components. Okay. So this one is autophagic digestion. Discuss in detail what is an autophagy. Autophagy in Greek word means self-eating. It is a catabolic process in which the non-functional cytoplasmic components and organelles delivers to the lysosome where hydrolytic enzymes digest them and allow cell to reuse them. So what it says that autophagy is a process in which the cell components or organelles that are the part of the cell is delivered to the lysosomes, where the hydrolytic enzymes of lysosomes are digest them, and the simplest form is reused by the cell itself. Like for example, mitochondria that are constantly being removed from cell via lysosomal system. It is the first get surrounded by membrane of smooth ER. So suppose this is our mitochondria. We know that mitochondria is constantly being removed from the cell system, and this is by process under lysosomal system which digests the mitochondria. So first it gets surrounded by the membrane of smooth endoplasmic reticulum. So this is the membrane of smooth endoplasmic reticulum. It is going to be surrounded like in this fashion and form a vacuole type structure. Okay, this. It forming a vacuole. This vacuole is known as autophagosome. This vacuole is known as autophagosome, which surrounded the cytoplasmic component, or we can say mitochondria. Okay, this one is autophagosome. This is further fused in the autolysosomes. The hydrolytic enzymes of lysosomes get discharged and mitochondria or other organelles get digested. They are getting digested inside the autolysosome. So this is the autophagy. Removal of dead cell. We know that in every day millions of cells are divided and regenerated, but our body maintains the number of cells. This is homeostasis, where the number of cells is stabilized and maintain the same number of cells by the body. Then what happened to the old cells? They get removed by our life savior lysosomes. Suppose look at this picture. This is an apoptotic cell. Okay, this is our apoptotic cell or old cell, which exposed some chemical signal on its surface. It's export the find me signal. Okay, which is recognized by receptor on a phagocytic cell. This is our phagocytic cell that is going to do the process of phagocytosis of this apoptotic cell. Finally, this cell, the signal that is present on the old cell or apoptotic cell having signal eat me, for example. So this is recognized by the receptors that are present on the phagocytic cell. This phagocyte may be macrophages. Okay. So finally this cell is engulfed by or phagocytosed by a phagocytic cell, which then transferred to the lysosome. So this engulfed phagocytosed phagosome is fused with the lysosome. It is getting fused with the lysosome. Okay. And all the hydrolytic enzymes that are present inside the lysosome, that is protease, DNase, lipase, glycosidase, helps in breakdown of this apoptotic cell. They are going to get breakdown into simpler forms which are used by this macrophage, and the some residuals that are still present, which are not of any use of the cell, is getting out from the cell by the process of exocytosis. So we can say that for maintaining the homeostasis, the old cell is engulfed by phagocytes via chemotaxis. So the dead cell then get transferred to the lysosomes, where lysosome containing a sufficient amount of hydrolytic enzymes, that is protease, DNase, lipase, glycosidase, which digest all the biological material of a cell. Role of lysosome in metamorphosis. In this process of metamorphosis, the amphibians and tunicates, the embryonic tissues like gills, fins, tails, etc. are get digested with the help of lysosomes, and the products are utilized by other cells for their growth and differentiation. So in metamorphosis process, some embryonic characters like gills, fins, tails that are present in embryonic stage are get digested with the help of lysosomes, and the products that are from the come from the digested parts are used or utilized by other cells for their growth and differentiation. Suppose in this example we are taking example of how the metamorphosis occurs from a tadpole to a frog. So in this picture you can see this is a tadpole having tail, okay, and in adult form this frog is not having any tail, okay, there is no tail present in the frog. This tail of tadpole is get digested by lysosomes, and the nutrients that are comes from the digested part is taken up by other cells for the growth of frog. So this is the tail of a tadpole which get disappeared of frog is due to the activity of lysosome, due to the activity of lysosomes. Okay, so lysosomes also helps in the process of metamorphosis. Lysosomes helps in fertilization. During fertilization, sperm's head contain lysosomal enzymes that secretes during fertilization, which help in penetration of sperm into vitelline layer of ovum. We know that so many sperms are in this race to fertilize the particular or single ovum, and only one sperm is able to fertilize the oocyte. So in this sperm we have the acrosome, and the acrosome contains lots of hydrolytic enzymes. The acrosome contains proteases and hyaluronidases and abundant acid phosphatase. This hyaluronidase helps in dispersion of cells around oocytes. So the hyaluronidases help in dispersion of cells which are around the oocyte, and the proteases break down the zona pellucida. The proteases helps in breakdown of zona pellucida. Okay, thereby forming a channel. So there is a creation of a channel or a pore from where the sperm nucleus penetrates to the oocytes. That's all for today, and if you have any query, please text me below and share your suggestions about the topics you want me to cover in upcoming videos. Thanks for watching.